Hello, everyone, and welcome to the second webinar of the APDA Connecticut Chapter Educational Webinar Series. My name is Marlene Argianis, and I'm the chapter coordinator of the American Parkinson Disease Association Connecticut Chapter. Thank you so much for joining us today. We are extremely grateful to Dr. Duarte Machado for volunteering his time to present today's topic of understanding the role of inflammation in Parkinson's disease. After some brief announcements, Dr. Machado will give his presentation and he will be answering your questions in a Q&A session. American Parkinson Disease Association Connecticut Chapter, or APDA CT for short, is part of the largest grassroots network dedicated to fighting Parkinson's disease and works tirelessly to assist the more than 1 million people with Parkinson's disease in the United States live life to the fullest. The APDA Connecticut chapter offers a variety of services in the state of Connecticut. Please visit our website to explore all that we have to offer. Our website also hosts a vast array of publications, webinars, articles, and information that can help you understand Parkinson's disease. If you are participating from another state, we have chapters and locations throughout the United States, and you can find that contact information on our website as well. APDA Connecticut is available through our helpline, email, as well as via our social media outlets. A special thank you to our educational webinar series sponsors. Our gold level sponsors are Abbott and Adamas. Our silver level sponsors are Abbey, APDA, Kiowa Curran, Savonian, and Supernus. And our bronze level sponsor is Brightview of New Canaan. Please visit their websites for more information about their products and services. Today, we are pleased to have representatives from two of our sponsors provide welcome remarks.
Hi all, my name is Argo Dalapati and I work with Abbott Deep Brain Stimulation. It's a pleasure to work with and sponsor America's Parkinson's Disease Association, the Connecticut chapter. I look forward to meeting you all in person soon.
Thank you, Argo. And from one of our silver sponsors, APDA, please welcome John France.
Good afternoon, Marlene, all distinguished members of the Connecticut APDA. My name is John France. I work for APDA Pharmaceuticals. It is APDA's honor and pleasure to be a sponsor this year, even in a tough virtual environment. The APDA does great work, and APDA is happy to have a long-term partnership over the last 5 years. Thank you for allowing APDA and myself to be part of this webinar opportunity. Thank you, Marlene.
Thank you, John. And now we will begin our presentation. We are delighted to welcome Janet Turcotte, a representative from our second gold sponsors, Adamas, who will introduce our guest speaker, Dr. Duarte Machado.
Hi, Marlene. Janet Turcotte. Thank you, everybody. Welcome. Our guest today is Dr. Duarte Machado. He is a board-certified neurologist with subspecialty training in movement disorders. He's committed to a career caring for those with movement disorders while a student at Crosby High School when his grandmother was diagnosed with Parkinson's disease. He is a 2001 graduate of Trinity College with a BA in neuroscience. He is a 2005 graduate of the University of Connecticut School of Medicine. He completed neurology residency training at the Yale School of Medicine, where he was also chosen to be a chief resident. Dr. Machado was subsequently recruited to join the faculty at Yale, where his primary responsibility was the care for patients with involuntary movements in the neurology outpatient clinic in the Division of Movement Disorders. Dr. Machado is currently Director of Program Excellence and Recognition at the Hartford Healthcare Ayer Neuroscience Institute, Chase Family Movement Disorder Center in Cheshire. Dr. Machado has participated in numerous clinical research projects, excuse me. He has been heavily involved in the teaching of medical students, residents, and fellows, and completed the medical education fellowship program at Yale. He serves as the medical director of the Connecticut chapter of the International Essential Tremor Foundation and on the American Parkinson's Disease Association Connecticut chapter board of directors. He has been repeatedly chosen as a Vital's top 10 doctors, awarded only to 1% of all doctors nationally who meet the criteria for this honor, and has been practicing medicine for 12 years. We are so excited to have Dr. Machado here with us today. Dr. Machado, welcome, and thanks so much for joining us.
Thank you, Janet, for that introduction.
You're welcome.
And to all of the participants for taking time out of your schedule to listen to this program today. And I thought that this topic is not one that's spoken about very frequently, but one that is important to review. So I want to start with this quote, that there appears to be sufficient reason for hoping that some remedial process may ere long be discovered by which at least the progress of the disease may be stopped. So this physician was referring to Parkinson's and referring to a disease process that again, if we can determine the intricacies and of how this disease process works and contributes to the disease itself, that we would have a way to stop this disease in its track. And so you may ask, well, who was it that came up with this quote? Which of your contemporary colleagues came up with this? And in fact, this was actually, let me advance it to the next slide here. So this was actually James Parkinson. So in 1817, so in the initial description of Parkinson's, there was already a thought that there might be a process, if we knew how it works, can in and of itself lead to us being able to stop the disease. And so what I hope to paint a picture of today is that that disease process may very well be neuroinflammation. So we'll start out by a brief overview of the immune system. It's composed of two branches: the innate immunity driven by macrophages, which in the nervous system, the main cell type is microglia, and this provides a first response. So these are the first responders when there's an insult, an infection in the body. Then the adaptive immunity is responsible for long-term memory responses. So the adaptive immunity is what makes antibodies to that insult, so that when the body sees it in the future, it can target it appropriately. And we know that there's not a single cause, but several causes leading to the common preferential loss of dopamine neurons in Parkinson's. So the initial thought of what causes neurodegeneration was this concept of oxidative stress. So one of the questions, for example, that came in even before the talk started was, why is it that dopamine cells are preferentially targeted in this neurodegenerative process? And in the 90s, there was a lot of research on oxidative stress. So in other words, the dopamine neurons, they have neuromelanin, that black pigment. There is an accumulation of iron in the cell. There's reduced defense mechanism against the generation of what are called free radicals. And then there's also dysfunction of the powerhouse of the cell called mitochondria. So here in this diagram, we can see that in the middle is iron, shown in the red circles. And iron is a cofactor for tyrosine hydroxylase to make dopamine and then to make its metabolites, but it also contributes to the generation of what are called reactive oxygen species, which essentially is like rust. So the cell with iron in it, it starts rusting. And this is something specific to the dopamine producing cells. So the dopamine cells were thought to be rusting and become stop working properly. And this is what resulted in ultimately their dysfunction. But there's something more than that. And so when I was a college student, this is what I was working on already with my professors was on experiments on reactive oxygen species. But in the last 10 years, the focus has shifted to neuroinflammation, and the trigger of which is still unknown. But the two most obvious culprits are the many different genes that have been described in Parkinson's, the first of which was in 1997, and then environmental factors, both of which impact on different cellular pathways that merge to cause dysfunction due to formation of a protein aggregate called alpha-synuclein. So alpha-synuclein is something that all of us have, whether or not we have Parkinson's. So alpha-synuclein is intrinsically important for cell-to-cell communication. However, when that alpha-synuclein starts binding to each other and clumping, and here it shows how it starts clumping and forming this fibril aggregate, this clump of proteins. In the picture above, we can see a cell body, a neuron cell, and long axon, and then the dendrites, the little branch points. And within the cell body are these clumps of these alpha-synuclein aggregates, also called Lewy bodies. So this is the pathological hallmark of Parkinson's. So it's whereby these alpha-synuclein proteins start clumping together. The body is unable to rid of them. They clump together, and they also cause cell dysfunction. So now the thought is, well, if these alpha-synuclein fibril aggregates are clumping together, what is causing them to clump together? What is that trigger that's driving this process? And the thought is that it's partly inflammation. So multiple studies report that the levels of alpha-synuclein in Parkinson's gut are higher than in the gut of those without Parkinson's. So there's been a lot of research on thinking about inflammation in the gut, since alpha-synuclein is present there. So to backtrack, the gut harbors trillions of microorganisms referred to as a microbiome. So the microbiome is important to help digestion of food, help with warding off harmful microorganisms, and also creation of needed vitamins. So the absorption of necessary vitamins and nutrients from the foods we eat is thanks to the microbiome. And we know that the microbiome in Parkinson's patients is different as compared to those without Parkinson's. So it's led to the theory that changes in the bacterial composition of the gut in people with Parkinson's may trigger increased inflammation in the gut, which then fuels the disease. So the earliest pathology associated with Parkinson's actually appears in what's called the peripheral nervous system. So the nerves outside of the brain and spinal cord. So these are nerves called the enteric nerves that serve our gut, the vagus nerve that connects our gut to the brain, and other peripheral nerves. And the thought is that alpha-synuclein that starts clumping in the nerve cells outside of the brain and the peripheral nerves then travel from the gut or from the nose into the brain. So that intestinal and systemic inflammation, alteration in the microbiota, and gut and blood-brain barrier dysfunction all contribute to Parkinson's disease pathogenesis. So this diagram illustrates how the gut is connected to the brain via the vagus nerve. And so the two conduits of entry into the brain are from the gut. So if there's inflammation in the gut, an alpha-synuclein misfolding, that'll then travel up the vagus nerve into the brainstem and then into the brain itself, or from the nose, the olfactory bulb and the entorhinal cortex, and then into the brain that way. So when we ingest or inhale some toxic chemical in the environment, coupled with our own innate genetic makeup, that could be the trigger, if you will, for then alpha-synuclein misfolding, inflammation, and then the start of Parkinson's. So why the gut and the nose proper? Because these two are very common sites of dysfunction. So for example, hyposmia, which is reduced sense of smell, or full anosmia, when patients have full loss of sense of smell, this affects more than 95% of people with Parkinson's. And Lewy bodies, those clumps of alpha-synuclein I was referring to, have been seen in the nasal epithelium, nasal cells, in the olfactory bulb, and as a very early manifestation. Constipation, also very common, early and late complaint in Parkinson's. In fact, both of these issues are common questions that myself and my colleagues ask of patients when we see them for the first time in clinic, when we're thinking about, could this person have Parkinson's? We ask them, any change in sense of smell or any constipation? If the answer is yes, that increases the pre-exam probability that when I actually examine them, then I'll find the physical manifestations of Parkinson's disease. So bowel frequency diverges from that of those without Parkinson's, perhaps 3 decades before the median age of Parkinson diagnosis. So people with Parkinson's may have constipation decades before they actually develop motor symptoms. The occurrence of constipation occurs before motor symptoms in 87% of people with Parkinson's. And there have been studies looking at patients who underwent routine colonoscopies and then later developed Parkinson's when they went back in a what's called a retrospective study of tissue from colonic biopsies. It showed aggregated alpha-synuclein from these, at the time, asymptomatic people decades before the onset of motor symptoms. So all these levels of evidence are pointing to the gut and the nose as portals of entry of inflammation into the brain. So an infectious agent targeting the gut and olfactory bulbs may be the initial trigger for Parkinson's pathogenesis. And although it is clear that a single organism is not consistently associated with Parkinson's, a Parkinson-like combination of motor symptoms known as Parkinsonism has been observed in the aftermath of infections in the past. And certainly a great example of that is the 1918 Spanish influenza pandemic, when we saw an encephalitis and subsequent to that, many people, 80% of whom had that type of encephalitis lethargical, who developed Parkinsonism. And of course, now with our current pandemic, there have been actually case reports of people who are asymptomatic, developed COVID infection, and have developed an acute Parkinsonism following that. And we know that COVID is associated with hyposmia or alterations of sense of smell as well. So how might viral infection result in dopamine depletion? One possibility is that the same direct viral invasion of the central nervous system and or a systemic, what's called cytokine storm or a burst of inflammation caused by the virus predisposes to dysfunction of dopamine neurons, as I explained. So too much of this inflammation then causes the alpha-synuclein clumping and causes reactive oxygen species to ramp up, and all of which drive dopamine dysfunction. It's also possible that the immune system response to certain influenza strains and other viral strains could produce a type of induced autoimmunity, resulting in the immune system driving their degeneration as well. So similarly, bacterial infection can also cause dopamine depletion. We know that there have been very exquisitely done studies looking at the activity of gastrointestinal microbes in relation to PD pathology. There was one study that looked at the stool samples of those with Parkinson's and those without Parkinson's and different subtypes of Parkinson's. So those who had akinetic rigid Parkinson versus those who had tremor predominant and postural instability type. And what the researchers found was that based on the stool bacterial composition, that the researchers could, with good accuracy, tell what type of Parkinson, well, not only that the person had or did not have Parkinson's, but also the type of Parkinson the patient had, based on their stool bacterial composition. So we know that there are changes in the gut microbiome that occur in Parkinson's, and that the overgrowth of some harmful bacteria and the lack of some good bacterial in the microbiome can shift the manifestation of Parkinson's in one way or another. We also know that patients with Parkinson's exhibit a greater frequency of H. pylori infection. So H. pylori infection seen in roughly a third of Parkinson patients, and those who have H. pylori infection is associated with a higher risk of developing Parkinson's also. So how is it that the gut bacteria can promote alpha-synuclein? Now this is complex, but certainly the gut microbiota promote inflammatory capabilities of microglia, the macrophages of the nervous system, through the production of short-chain fatty acids. This inflammatory environment enhances alpha-synuclein aggregation, and then microglia are further activated. This promotes a cascade event that leads to additional alpha-synuclein aggregation and propagation in the gut. And so distinct microbes associated with PD rather than general microbial stimulation manifests as the disease symptoms. And so here we have this way of thinking about Parkinson's now, which is more complex than what we initially thought, certainly. And this diagram was just published last month in the main journal for movement disorder specialists called Movement Disorders. It was in the January 2021 issue, on the cover of the issue, to highlight its importance. And that this is showing environmental factors that disrupt the homeostasis, increase risk of Parkinson's. So that's bacteria, viruses shown in green, environmental toxins that not only cause microbiome changes and neuronal damage, but also potentially can impact gene expression, cause a cytokine or inflammatory release, proliferate other cells in the called T cells that can then cause self-directed autoantibodies to occur to further cause neurodegeneration, inflammation, and systemic infection. So it's all of these coming together. It's not just one pathway, and that's been part of the challenge of developing a treatment option that's going to transform the treatment paradigm for Parkinson's. It's complicated. It's not just a single pathway. So environmental factors, chemicals, viruses, bacteria may disrupt the immune homeostasis, as I mentioned, not only by directly causing tissue damage, which I've reviewed, but also altering immune cell function in this concept of molecular mimicry. So in the brain, the affected neurons might activate microglial cells, and alpha-synuclein also activates microglia. So it's not just then the initial trigger, the trigger starts it, but then the end results of that, the microglia and the alpha-synuclein, then they in and of themselves trigger other actions to occur. So the degenerating neurons, once they start breaking down, the debris of these neurons then also activate further microglial cells. So it's like a vicious cycle that can occur where there's chronic and progressive neurodegeneration. So you have the pro-inflammatory cytokines causing neuronal degeneration and breakdown. Alpha-synuclein aggregates are released from the cell. They cause a reactive gliosis in the brain, further inflammation, neurotoxic factors, and it's this vicious cycle that occurs with multiple points along the cycle. So the goal of clinicians and researchers of the future will be to continue to target these points in this cycle to try to break this cycle. So what perpetuates neuroinflammation? Again, misfolded alpha-synuclein can actually transmit from one diseased neuron to a healthy one. So that the aggregated alpha-synuclein protein promotes normal alpha-synuclein to then misfold also. And this causes a chain reaction process known as a prion conformer reaction. And this is it's a hard concept to understand. And if you would need to go back after to review this webinar once more, it'll be, it's being taped so that and posted so that you can go back later to view this once more. Because it might take more than once to understand this concept fully, but this concept has been around for about a decade now, whereby, again, alpha-synuclein, the misfolded protein in some cells can trigger other alpha-synuclein to also misfold and lead to this cycle of inflammation and neurodegeneration. So here it shows that concept that whereby the one cell has the alpha-synuclein that's taken up, and then it starts combining to form this clump of protein called a Lewy body. And then once a Lewy body is released by a cell being broken down, neighboring cells then take up that misfolded protein into themselves, and they become infected. So what's meant by a prion is a protein that appears like an infectious particle. It can go into and enter other cells and induce changes in the cell, just like an infection would. And that's thought to be happening in the neurons in Parkinson's also. So stopping the inflammation early before multiple cells become involved is an approach that will likely need to be taken in the future. So there were also questions about, well, what happens if I already have an inflammatory condition independent of Parkinson's? So we know that when a systemic inflammatory event is combined with chronic neurodegeneration, that these microglia of the brain are further activated and damaged endangered neurons, accelerating the pathology. So for example, inflammatory bowel disease increases risk for developing Parkinson's, not only because Parkinson's and IBD share a genetic linkage through a gene called the LRRK2 gene, but also because the inflammation that's part of inflammatory bowel disease further accelerates the inflammation and neurodegeneration. So studies of neuroinflammation in Parkinson's quickly gaining speed in the field and promise to open door to entirely new approaches to neuroprotective therapeutics. And that's what we need in Parkinson's. We have many good ways of managing symptoms now, but we don't have a neuroprotective therapeutic other than exercise at this point. So an important aspect of these approaches is that it is possible that a therapy for Parkinson's might actually be based on existing immunomodulatory treatments, such as those that have been used for other inflammatory disorders. So in one large population-based study, the use of immune suppressants, namely corticosteroids, and a category medication called inosine monophosphate dehydrogenase, IMDH inhibitors, was associated with a lower risk of Parkinson's. So the IMDH inhibitors are medications like azathioprine, mycophenolate, and they work by suppressing T cell activity, which could suppress peripheral alpha-synuclein aggregation in the GI tract. So again, trying to halt inflammation and that protein aggregation in its tract in the periphery before it gets into the brain and does damage. Now, why hasn't an immune therapy been used in Parkinson's yet? That's because an unmet need is that we do not yet have a reliable approach to study neuroinflammation processes in living patients as the disease changes and evolves. So the main approach is so far to study blood and spinal fluid and brain imaging. So we know that studies have shown elevations of certain pro-inflammatory markers in the blood, TNF, IL-1, IL-2, IL-10, and this has been associated with faster progression of symptoms. And also, we're moving into the realm of being able to do brain imaging and looking at inflammation. So there's a particular neuro tracer called TSPO, a translocator protein that's predictive of neuroinflammation also. And I've been involved in PET imaging using the TSPO in looking at candidates for drug therapeutic candidates for Parkinson's. And so this is a field that's evolving, but we're still learning more about what is the optimal way to manage inflammation that's also reliable and reproducible over time. So there's been some success with each of these approaches. The overall strategy of monitoring inflammation in living patients with Parkinson's is still lacking. So we need to really define and refine the approach to monitoring this and do this consistently in clinical practice. So this is going to be critical for us to understand the causal link between the progression of inflammation, neurodegeneration, and alpha-synuclein spreading from one part of our body, from the gut and elsewhere to the brain. There's also been numerous studies evaluating whether the use of anti-inflammatory drugs is associated with reduced risk of developing Parkinson's. A lot of studies have been looked at use of NSAIDs, for example, the non-steroidal anti-inflammatories. In another category of looking at anti-inflammatories, one could say, well, how about use of probiotics? Use of probiotics has been shown to ameliorate constipation, Parkinson's, and so what we need to understand is what's going to be the interaction between the bacteria from the probiotic that's introduced into the system and the innate bacteria that's there with regards to the immune response in Parkinson's. So that still needs to be elucidated. So, well thought out clinical trials using very specific probiotics to introduce the good bacteria that's lacking in those with Parkinson's to try to offset that balance between good and bad bacteria. So ultimately, what we have in Parkinson's is, again, the immune system dysregulation, inflammation, leaky gut with release of cytokines, and then bacterial overgrowth causing alpha-synuclein aggregates in the cells of the enteric nervous system, that peripheral nervous system, that then travel up the vagus nerve into the brainstem, into the brain, which then leads to those alpha-synuclein aggregates essentially transfecting the cells in the brain and causing alpha-synuclein aggregation in the cells of the brain, in particular the dopamine producing cells, because they're so vulnerable to this. Going from that state to something like this, whereby we improve the function of the gut through modulating the immune system microbiome through probiotics, prebiotics, symbiotics, other phospholipid precursors to try to improve gut function so that the alpha-synuclein does not clump and misfold and infect the enteric nervous system cells, so that the alpha-synuclein does not go up the vagus nerve into the brain and cause neurodegeneration. So this is something that we hope can be accomplished in the future. So now we'll move into diet. Is there a way that we could do something now? So when thinking about diet, and this was another popular question prior to the start of the webinar today, there's several different diets out there. The Western diet, of course, is notorious. That's our diet. Notorious for its high levels of processed and fried foods, sugar, and red meat. The Mediterranean diet, characterized in part by being antioxidant-rich, with vegetables, whole grains, fish, and certainly much reduced meat and dairy. And then there's the Mediterranean DASH intervention for neurodegeneration delay diet, the MIND diet. So this diet uniquely rewards leafy green berries and poultry intake. When they study these different diets in those with Parkinson's, a higher adherence to the MIND diet was significantly associated with a higher age of disease onset, especially so in women who had a difference of up to 17.4 years between the highest and lowest tertiles of diet adherence. So those who adhered to the diet most stringently delayed the onset of Parkinson's by 17.4 years for women. I mean, it's just so remarkable that this type of change was seen. So we know that dietary modification can have a big impact on the development of Parkinson's and potentially the time course of Parkinson's. So I know in my practice, I ask a lot about diet because it is so important, not only with regards to gut inflammation, but also other things like even management of constipation. Since constipation is so common in Parkinson's, dietary modifications can help improve constipation. But certainly future work will need to investigate the effects of the MIND diet on disease progression, inflammatory markers. Again, once we know how to monitor inflammation well, this will be part of the standard clinic visits to monitor these inflammatory markers and also to better understand the gastrointestinal symptoms such as constipation and dysbiosis. So leafy greens and berries, which are specific to the MIND diet, are rich in antioxidants such as carotenoids, flavonoids, folate, and vitamin C and E. And some or all of these have been associated with the lower Parkinsonism risk and reduced disease progression in both animal models and human cohorts. So given all of this data, in our practice, we're very much following the model of monitoring a patient's diet and assessing key vitamins and nutrients. So we tell our patients eat a rich diet in fresh vegetables, whole grains, healthy oils, limit dietary intake of dairy, red meat, and sugary processed food. At least once a year, I send my patients for a panel about vitamin B1, B12, D, E, and folate, and then aggressive management of constipation. The goal is a bowel movement within 48 hours. So by doing these things, it's at least an initial step to try to improve gut function and try to reduce this cascade of events that I've described today with regards to inflammation starting in the periphery and ultimately driving the process of neuro-inflammation and then neurodegeneration within the brain. So final thoughts are, there's multiple factors that trigger Parkinson's, both genetic and environmental, but inflammation may represent a common pathway by which the disease progresses. And if this idea is correct, then immunomodulatory treatments may prove valuable across the whole spectrum of Parkinson's. And one day, controlling the immune system, I think, will be part of the prescribed course of treatment for Parkinson's. So we'll be using just symptomatic treatments with dopamine, but also trying to treat the underlying inciting factors, not just, again, replenishing lost dopamine. So at this time, I'm happy to take questions, and we have some time for questions.
Okay, hi everyone. Thank you so much, Dr. Machado, for this informative presentation. To introduce our guest Q&A moderator, please welcome Chad Wickland, who is a representative from one of our silver sponsors, Supernus. Chad?
Thank you. I am delighted to introduce our guest Q&A moderator, Dr. Rebecca Gilbert, Vice President, Chief Scientific Officer at the American Parkinson's Disease Association. Dr. Gilbert is responsible for overseeing APDA's research portfolio in conjunction with the APDA's Scientific Advisory Board, and also provides medical and clinical expertise to support APDA's educational programming and digital and printed content. Welcome, Dr. Gilbert. Thank you.
Thank you so much for that introduction. And thank you, Dr. Machado, for that really informative and excellent talk on an extremely important topic to many people listening today. We have a number of questions. Some have been submitted ahead of time and some that are coming in as people have heard your presentation. So we'll try to answer as many questions as possible. That may not be possible in the time we have left to the presentation, but we'll do our best. So one question from Frank is to shed some light on this topic of inflammation and Parkinson's. Is there a higher prevalence of Parkinson's in people with autoimmune conditions?
There is, yes. So I've highlighted the inflammatory bowel disease. We know that there are other autoimmune conditions like celiac disease, which also affects the gut and for which we do see a higher incidence of Parkinson's. And so in the future, I believe we'll be screening more often for autoimmune conditions that can be seen in conjunction with Parkinson's. And so Parkinson's really is best now managed in a multidisciplinary way, whereby we're really in a collaborative arena of care with our other colleagues in other fields of medicine, since we know that that there is association of Parkinson's with these other conditions.
So on a related note, someone in a registration asked, can you try to distinguish between inflammation that someone may experience in their joints or in their body versus inflammation in the brain? Are those two related? If you have inflammation in your body, do you necessarily have inflammation in your brain, or are they related or not related?
Yeah, it's a great question, and that's the challenge. Like we just don't have a good way to tease out inflammation in one site versus the other. There is a protective, the brain is protected to some degree from external inflammation through what's called the blood-brain barrier. But we know that there are other means, as described, whereby inflammation can enter into the central nervous system and bypass that blood-brain barrier. And we just don't have a good means of assessing for inflammation in the brain proper. And certainly we're not advocating for doing spinal taps on all of our patients to assess that. So we have to have better means of really gauging what happens in real time with regards to inflammation in the brain when someone may experience a flare-up of inflammation in their body.
Okay, excellent. And you mentioned a study or studies that looked at NSAID use, non-steroidal anti-inflammatory drug use. Were there any conclusions in terms of how a person with Parkinson's should deal with NSAIDs? Should everyone be taking NSAIDs? What are the conclusions that you draw from that?
Yeah, so the data's been mixed. So the studies looked at patients who, for example, had osteoarthritis for years and were taking NSAIDs to treat that peripheral inflammatory response in the joints. And then they looked at, well, patients on NSAIDs, what happens to them over time? Are patients who take NSAIDs less likely to develop Parkinson's compared to those who don't take NSAIDs? And while some studies showed that yes, others didn't show that association. And it gets more complicated from there because there are subsets of Parkinson's that seem to have more of a protective response. So in other words, those who took NSAIDs who had a LRRK2 mutation were less likely to develop Parkinson's compared to those who had a LRRK2 mutation and didn't take NSAIDs. They were more likely to develop Parkinson's earlier. So NSAIDs seem to help to stave off the condition compared to not taking NSAIDs. But then the flip side of that is that NSAIDs chronically can have other problems in and of themselves with regards to its effect on the gastric lining and cause ulcers. So the data is still mixed. While some people may have seen the benefits of NSAIDs with regards to delaying the onset of Parkinson's, the actual story is that it's a lot more complicated than that.
Okay, fantastic. I think that's what we're learning. Everything is much more complicated than we hope. There have been a few questions about the use of turmeric. So I'll expand the question a little bit. You mentioned specific diets that may be helpful to lower levels of inflammation. But are there specific products beyond a general diet that you recommend to your patients?
Yeah, so this has also been evaluated recently with regards to studies looking at use of a variety of different supplements in different populations. So worldwide, in different countries, to say, well, in countries that where the populations are more likely to use a particular type of supplement more routinely, tracking those patients. And the conclusion was that there was enough of a strong signal to indicate that a particular supplement was neuroprotective, if you will. So based on that, what I tell my patients is that if they wish to use a supplement and there's no contraindication to doing so, that might be an antioxidant. then certainly I have no objection to that. At the same time, it's challenging to monitor how much benefit that supplement is having, of course. And then there are supplements that can be expensive, that might be proprietary, as a combination of different antioxidants that patients may be interested in taking, when again, we just don't have enough research to back a particular type of antioxidant or supplement over another. So that's why my approach has been to check levels for things that we can check for, and then supplement accordingly. So we supplement based on what an individual person's body is telling us that they need. So even when it comes down to, so for example, vitamin E, vitamin B12, not every person with Parkinson's will have a deficiency, but then some do. And when we can supplement, it does have an impact on their energy level, on their overall well-being that they report back to us. So by doing directed supplementation, I think that's more likely to have benefit than outwardly recommending one particular type of treatment like turmeric for everyone. And that's why we haven't gotten to that point where we're recommending a supplement for every single patient with Parkinson's.
Thank you. That was a wonderful answer. We have a great question from Frank here. Is the progression of Parkinson's from early to advanced correlated with increased inflammation?
Yeah, that is a great question. So the general thought is that yes. So as Parkinson's progresses, we see greater dysfunction in the gastrointestinal tract, more difficulty with having regular bowel movements. Patients go longer and longer between bowel movements, adding more and more over-the-counter and prescription-based agents to manage bowel movements. And then when we check inflammatory markers, we do see that they are more likely to be increased over time. And so certainly disease progression is correlated with more inflammation by the measures that we have. Of course, we need better tools of monitoring that so that we can develop clinical trials that will help us to better evaluate interventions with regards to progression. But the short answer is yes, that more as disease progresses, there is more inflammation.
Thank you for that. We have another great question from Jennifer. Do you recommend, or what is the importance of organic produce versus regular produce? Is that something you recommend to your patients or not?
Yeah, so ultimately, the whole issue with the environmental toxins and pesticide use is just that in foods that are not organic, that there's always the potential that they're processed in a way that might introduce some impurities or some toxicity to them, because a case in point is dairy. Why is dairy associated with worsened Parkinson's? And that's because of the fact that in milk production, and there's a great study showing this out of Hawaii, where there was a cohort of people from one of the islands of Hawaii that had a higher incidence of Parkinson's compared to another cohort, and they actually traced it back to milk, where the source of milk was coming from. From one place, it was being produced and manufactured organically, and another it was not. And there were some pesticides that were detected in the milk source chronically for as long as it was produced at this manufacturing plant. And that led to this disparate rate of development of Parkinson's in this controlled environment of the different islands of Hawaii. So introduction of these toxins and pesticides into the food sources and food sources that are not organic certainly can have risk. Of course, organic foods are more expensive and not easily accessible by everyone, especially, there's a lot of discussion about food insecurities in the time of pandemic. So we tell patients, to do the best that they can with regards to accessing foods that they can afford as healthy as possible.
Fantastic. You mentioned earlier in your talk about H. pylori and its potential role in this entire discussion. So the question is, if you treat H. pylori, do you help people with Parkinson's disease? Is that something people should be tested for?
Yeah, so another great question. And this has also been looked at quite a bit with regards to patients who have, in particular, like gastroparesis, where they feel like this sense of bloating and sense of where they don't feel like they're emptying their stomach well, or they have ulcers, gastric ulcers, or burning. It's easy enough to test for H. pylori infection through a stool sample or through a blood test. And if present, there's a described antibiotic course, a triple or quadruple antibiotic course that can be prescribed to patients, which may certainly help improve bowel function and thus reduce inflammation. So it's part of breaking that cycle of inflammation. So for my patients who do have those concerns, then I will send them for H. pylori testing in order to then try to eradicate H. pylori infection and help to restore a more normal microbiome.
Fantastic. On those lines about the microbiome, we had a few questions about the proliferation of good and bad bacteria in the gut. Is there a way to know whether your microbiome is a good one? Is there a way to test for that and to potentially change something about how you interact with the environment to improve your microbiome?
Yes. So I've started doing that, like obtaining stool samples and testing for that, and depending on the insurance. So for Medicare, pure Medicare, not managed Medicare, but for pure Medicare, there are some companies that will do just that you described. So one is called Genova, whereby I'll give patients a kit to take home with them. They'll collect the stool sample, they'll ship it out, and then I'll receive a report that will indicate, is there a bacterial overgrowth? Is there an enzymatic deficiency? Is there inflammation in the gut? It does give us a rich sense of gut health that way. The next step is to say, well, now, how do we modify that? Certainly if there's an enzymatic deficiency, like a pancreatic enzymatic deficiency, then that can be managed. If there's a gut dysbiosis, that's a little bit more tricky, because again, what we don't have is a, and we may in the future, but a probiotic that's specific for Parkinson's. So generally speaking, I advocate for a probiotic that is variety. So it contains a variety, like at least 30 or so different types of bacterium, and then also one that's potent. So one that's like 50 billion colony-forming units or greater, because we know that the more common over-the-counter variety of probiotics, like a line, is just a single strain of a probiotic and not very potent. And so that might not be sufficient to overcome the dysbiosis of Parkinson's. But even amongst the readily available over-the-counter probiotics, they may not specifically help to restore the gut microbiome in a way that needs to be restored in Parkinson's. Now that there have been studies, and several of them, to conclusively say these are the fingerprints, if you will, of the gut microbiome in someone with Parkinson's. This is what the microbiome looks like. And so with Parkinson's, this is what someone without Parkinson's look like. How can we replicate that? Can we develop a probiotic concoction that is very specific for Parkinson's? I hope that that's the goal of the future.
Great, fantastic. Amazingly, we are nearing the end of the hour. I think this is going to be our last question, unfortunately. We'll see if we can maybe squeeze one more in. There have been a number of questions about other gut problems that people have with Parkinson's? You mentioned constipation a lot, but people have commented about bloating, about loose stools. Can you comment more generally about gut and Parkinson's disease? And are those also measures of inflammation? How does that feed into the inflammation process?
Yeah, so if patients are also alternating between loose stools, then that's where I'll involve my gastrointestinal colleagues, my GI colleagues, to evaluate, is there some other type of pathological process within the gut? So I also mentioned about celiac disease. So is there something else that's causing looser bowel movements that needs to be evaluated? And if it's something like celiac, that is something that's treatable, that's modifiable, altering the diet even further by going on the gluten-free diet. But certainly if it's more complicated, I think doing a stool analysis, referring to GI, evaluating for H. pylori, some other infection is certainly important. And then seeing what other medications the patient might be taking that might be contributing to looser bowel movements. But keeping the gut healthy is something that is a big takeaway message from this talk. Because of the importance of it for brain health too. Like that gut-brain connection is there, supported by a lot of research, and so it's really important that we keep our guts healthy.
Fantastic. And I think we do have time for one last question, which highlights an important issue, which is some of the things you talked about to potentially decrease inflammation, maybe diet and whatnot. Does it matter whether the person has been diagnosed with Parkinson's already or not? Would these interventions be more useful prior to Parkinson's being diagnosed, or is sort of the horse out of the barn once Parkinson's is diagnosed already? And how do you consider that dichotomy?
Yeah, I think that it's never too late to help reduce inflammation and help to reduce some of that inflammation that's present. So even in those of you who have more advanced Parkinson's, I think it's still worth, if you haven't already, to really evaluate your diet, evaluate whatever could be causing some inflammation in the system. Because I did mention that in the setting of some other inflammatory problem, that there's an acceleration of neurodegeneration or at the very least worsening of Parkinson's. And we know this so well from our patients who, for example, develop an infection, a urinary tract infection. Even after the infection is cleared, those with Parkinson's just seem to take a little bit longer to rebound back to baseline or following a hospitalization. And one can say, well, it's because they were deconditioned, they weren't as mobile, and that's why it's taken a bit longer for them to come back to baseline, or even cognitive function and how inflammation can impact on cognition and worsen cognitive function. So while these other aspects of well, it's the infection itself or the deconditioning may be true, one could also say, well, it's the fact that there's been a surgence of inflammation in the body as a result of this secondary infection. That's what's driving inflammation. So, so the bottom line is, it's never too late. Start with making those dietary changes, requesting check of vitamin levels in your blood to see whether or not there's any deficiencies that need to be corrected to improve the body's immune system and fight against inflammation. And then also, if there's a significant GI disturbance, seeing a gastroenterologist or having your neurologist work that up further as well, in order to fully understand the full spectrum of inflammation in the system.
Fantastic. Thank you so much for all those answers. I think we are officially out of time for questions, so I will pass the conversation to Marlene to deliver some final remarks.
Thank you, Dr. Gilbert, for being our guest moderator for the Q&A session And thank you so much, Dr. Machado, for a wonderful presentation. For additional information, answers to your questions, and resources, please visit our website. You can also email, call us via the email and phone number on the screen. The last session of the APDA Connecticut Educational Webinar Series will be held on Monday, March 22nd, 2021 at 2 p.m. Eastern Standard Time with guest speaker, Dr. Michelle D'Agostine, who will be presenting on non-motor symptoms in Parkinson's disease. Registration is now up on our website. Please save the date for our annual educational symposium. Unfortunately, due to COVID, this year's event will again be virtual, but we look forward to a very informative week of presentations on various topics related to Parkinson's disease. The week-long event will take place from April 26th to April 30th, every day at 2 p.m. Eastern Standard Time. Additional information and registration will be up on our website very soon. If you enjoyed today's webinar, we hope you will consider supporting APDA Connecticut with a donation. With your help, APDA Connecticut can deliver more programs and services like this one, which are needed now more than ever during these very challenging times. On behalf of the entire APDA Connecticut chapter, many thanks to Dr. Duarte Machado, Dr. Rebecca Gilbert, all of our participants, and our sponsors, Abbott, Adamas, Abbey, Acadia, Kiowa Curran, Savonian, Supernus, and Brightview and New Canaan for supporting our program. If you know someone who missed today's program, if you joined late, or if you would simply like to watch again, you can view a recording on our website at apdaparkinson.org/CT within the next day or two. We will also be posting any questions that weren't answered today. Thank you, everyone, and have a wonderful afternoon.